Yeah. Good morning and welcome to our CoE seminar from a very chilly Johannesburg, who is currently somewhere around 2 degrees Celsius. Our speaker this morning is Professor Simon Mukwembi, the current head of school of the School of Mathematics at the University of Witwatersrand. He was previously chairperson of the Maths department at the University of Zimbabwe, and he began his academic career at UKZN. He works in graph theory and some of the applications, but that leads us to today's talk, where he will be talking about pain, graph theory, Alzheimer's disease, and how all of these things link together. Thank you, Simon.
Thank you very much, colleagues. Thank you very much, Prof. Watson, for your kind words. Thank you, colleagues. Thank you all for coming. All those online, good morning, and thank you for attending. Perhaps let me start by thanking the Center of Excellence for affording me this opportunity to make a presentation. But not only that, but I would also like to thank them for funding some of my projects. And what I'm going to present is also going to touch on the projects that the center is funding. I remember, well, a long time ago, when I was doing high school, perhaps lower grades, my mother was always advised to make sure that I don't do maths at school. The community was so nervous about their children being exposed to simultaneous equation. They hated them. People were thinking that it's all theoretical. If you are to do maths, it's just theoretical. Even we find it when we apply for grants, it's quite difficult to get grants. Why? Because they think mathematics has no applications, it's just a theoretical subject. But I'm really grateful for the center for providing, the funding. And today, as Prof has said, I would like to talk about maths, precisely graphs. And we all experience pain one way or the other. I will also talk about pain. I will also talk about Alzheimer's disease, something that we worry about, especially when we get old and, we become prone to that. So I'm going to talk about that as well. So just to summarize, I'll talk about graphs, and within graphs, I will worry about a special parameter of these graphs. That parameter is actually based on the degree sequence. And then we move into pain, and then we move into Alzheimer's disease. So let's start. What is a graph? Most people would always think that a graph is that of sine. A graph is that of cos. In our context here, that's really not what we call a graph, but rather a graph is just a collection of points, some of which are linked for purposes of communication, the purposes of transferring that is diffusing in the network. You would find that the world that we live in is highly interconnected. It consists of objects. Some of these objects might be connected, and therefore a graph actually provides a natural framework that represents such. So when you look at what we call a graph, you will see that we've got those dots, and we call those vertices in graph theory. And we've got these lines that are joining some of the vertices. We actually call those edges. So from time to time, you will hear me saying, oh, a vertex or an edge. Those are the things that we'll be referring to. If you look at this graph, you can see that it has 5 vertices. So the order of that graph is actually 5. If you look at this graph, you will see that it has 1, 2, 3, 4, 5 edges. The size of the graph is 5. Perfect. What do I mean? Let me just give you a very quick example on where we use graphs. In sociology, they actually use graphs to represent a community with the sexual relations in the community. So over there, when they use graphs in sociology, they say each human is represented by a vertex, and where there are sexual relations, they represent that by joining the corresponding humans. So for example, this human being here is sexually related to 3 other individuals. This might actually be a wife of a very, popular husband, who might even be a professor. The world was shocked. Sometime in 2002, the world was shocked when this research came out. That was actually a sexual network at Jefferson High School in the States. And these are the connections. You can see that the pink, those are the females, and the males are the blue. So you can notice this one here. This girl had actually 1, 2, 3, 4, 5, 6 romantic relationships during the 6 months period. And we can't leave this slide without noticing this guy over here. You can lose count. So this brings out exactly what happens quite clearly. So formally, our graph, we said, it's a structure consisting of what we call vertices and edges. More important is the fact that for each vertex, we've got a number of measures that we can actually characterize it with. One of such indices will be what we call the degree of a vertex. The degree of a vertex is simply the number of vertices that vertex is joined to. So for instance, if you look at vertex U here, its degree is actually 3y because it's joined to Q, it's joined to Y, it's joined to Z by an edge. So its degree is 3. The degree of vertex V is actually 1 because it's joined to W. And in that case, we say W is a neighbor of V. Why? Because they are joined. Now, we now know how to calculate degrees of vertices. The degree sequence of your graph is simply a list of the degrees of the vertices of the graph. But in mathematics, we then say, let's be methodological. We often then write the degree in decreasing order or in non-increasing order if you want. So we start with the higher degrees first and going down. So for this graph, yes, we have U has degree 3, W has degree 3, Y has degree 3, Z has degree 3, and we have Q and V having degree 1 and 1. The smallest of these degrees is what we call the minimum degree of your graph. So for instance, in this case, our minimum degree is actually 1 because our smallest degree is 1 over there. Now, a very important parameter of a graph is what we call the diameter of a graph. We are referring to here the largest distance between pairs of vertices in your graph. And by distance, we really mean to say, if you have vertex U and vertex V, how many paths or how many routes do you have to move from your V to your U, or from your V to your Y? How many routes do you have? In each route, you count the number of edges that you traverse. That's what we call the length of that route. But our worry, because we want to be economic, we will consider the one that has a shortest length. And we call that the distance between your V. Look at a situation whereby we are modeling cities in a town. Sorry, we are modeling cities in a country. You look at Joburg, you look at Cape Town, and you say, what is the distance between Joburg and Cape Town? There are quite a number of routes that lead from Johannesburg to Cape Town. But we are more worried about the economic one, the shortest one. That is what we then call the distance between your Joburg and your Cape Town. So for instance, in this example, we have V and we have our Z. We have quite a number of paths from V to Z. Here is one, you move from V to W, then to Z. Here is another one from V to W to Y to Z, or from V, W to Y and to V, and then back to Z. But you can see that a shorter one is this one, from V to W and straight to Z. So then we say the distance between V and Z is actually a 2. So as I said, the diameter then is going to look at which pair in the graph is having the highest distance. So for instance, in this graph, you can see clearly that your U and your V are furthest apart. That's the pair that is furthest apart. And so the diameter is actually the distance between your V and your U. You will see that this parameter always occurs, and it has a lot of applications. So one of the things that we worry about is, given a network or given a graph, what is its diameter? That question is often difficult to answer, especially if your graph is very large. And practically our graphs are large. So people then say, well, we can't really compute this, but we can at least give some bounds. And these bounds actually help a great deal. The first question that one will ask is, whenever you are given a graph of order n and minimum degree, say delta, how large can the diameter be? Well, a lot of people worked on that and independently discovered that your diameter will never exceed 3 times the order divided by delta plus 1, plus some very small constant. And this was actually proved. In graph theory, we sometimes ask, how good is this bound? Wouldn't it be a bound that's trivial and that is not even achievable? In other words, is there a graph G with diameter exactly this quantity? If there is such a graph, then we are happy. We know we can't improve this bound anymore. We know our result. So the question is that bound best possible? How good is the bound? It turns out that yes, the bound is best possible. And you can see that this kind of long sausage graph with degrees that have almost the same size, almost of the same quantity, which we then call a regular graph. You can see that this graph here actually has the biggest diameter. And its diameter, if you calculate it, you will see that it's exactly almost this quantity. So that bound is sharp. But do take note that there is a lot of regularity in the degrees of the vertices. This graph here is almost regular. The graph is almost regular. For practical purposes, you wouldn't really want to say all we have are regular things. No, things differ. And for that reason, people ask the question, how do we define irregularity in graphs? How do we define irregularity in graphs? This group of researchers actually said we will just define the opposite of irregularity. The opposite of regularity in graphs. And this is how they defined it. They said a graph is highly irregular, provided it is connected. In other words, it's in one piece, and that each of its vertices are adjacent only to vertices with distinct degrees. For instance, if you look at this graph here, this vertex is adjacent to a vertex of degree 2, and a vertex of degree 1, and a vertex of degree 3. And all other vertices are like that. This is what they call highly irregular. More research took place. They actually showed that this class of highly irregular graphs exist, and it's actually a very big class. But the concern in what I'm going to talk about today is regularity isn't really the practical thing. We also have irregularity. And the question is, in light of what we have introduced, which is the opposite of regular graphs, have we done enough to answer the question, how do we define irregularity in graphs? It is also a question that occupied my mind for a very long time. And in 2012, I re-asked that question, but differently. I said, how far or near is a graph from being irregular or regular? Then I coined this concept of irregularity index of a graph as a measure of what we have asked there. So the irregularity index of a graph is simply the number of distinct terms in that graph's degree sequence. Simply the number of distinct terms in that graph. And then the next question was, if you look at the extremal graph for that bound on diameter, you will see that it's almost regular. In fact, if we are to qualify it using the irregularity index, which is the number of distinct terms in its degree sequence, you will see that the index is actually 2. Here you have vertices that are of degree 5 or 6. So the irregularity index there is 2. That was our extremal graph. So we then ask the question, if in addition I know irregularity index, can I improve that bound on diameter? The answer to that question is yes. You can actually do better. You can improve that bound. So this is one of the results that I proved. And is that bound best possible? The answer is yes, it is actually best possible. And you basically replace the end part of the our previous extremal graph by a graph which defines the irregularity index that you have in general to be your T. I just hope that this is not painful and this has not been painful. For that reason, I will turn a little bit, show the connection later on. But as I said, one of our concerns is pain. There is general agreement that there is a huge rise for the need of painkillers. That has actually increased substantially. And some other researchers actually looked at South Africa in particular. And they said 1 or more in every 5 South Africans has chronic pain. This was in 2023. And it's a big concern. Now, we even have more problems. The existing painkillers actually suffer a host of side effects. For instance, one of the painkillers, rofecoxib, was actually withdrawn, according to this researcher, why it had caused tens of thousands of deaths. And some said, we know what we need to do as a world. we really need to develop relative safe and effective painkillers. When you say develop, bring to the market, what does it involve? You will see, colleagues, that this is the drug development process. First, we have an infinite pool of chemical compounds that we need to select from that will work. So the first stage involves selecting the best, and then you get into preclinical trials. For the ones that you have said, we think these ones will kill pain, right? You will take them for preclinical trials. That will involve two things. Take them into the laboratory, you do the test, and you get your data, you see whether they work, and you take them for animal studies. The Center of Excellence is actually funding one of my projects where we are taking the painkillers for animal studies. Then from there, once all that is successful, you go for clinical trials. First phase in the clinical trials, you take a group of people between 20 and 80 in size, and you administer the drugs. And if you are successful, you go on to phase 2, where you increase the number of participants between 100 and 300, and then you move on to phase 3. Then after that, you'll be talking about getting close to a registration. So this is a very long and risky road. So the question is, the biggest question, especially when you look at the initial stages, which will take between 3 and 6 years, the biggest question is, from this pool, an infinite pool of chemical compounds, how do we identify or even design new ones that have got certain desired properties, even design compounds that have got certain properties? For instance, you want them to work. I spoke about side effects. You don't want them to be highly toxic. You want them within a good range. Let me point out from the mathematics side that as early as 1875, Arthur Kelly, a British mathematician, actually observed that, you know what, if you look at a chemical compound, you can represent it as a graph. You can model it as a graph. And how did he propose? He said, look, a chemical compound has atoms, it consists of atoms, and some of these atoms are bonded together. So he said, it's very simple and clear. All you do is, wherever you have an atom, you put a ring. And wherever you have a bond between the atoms, you put a what? An edge. And here is one simple illustration. If you look at the chemical compound, aspirin, that we take to kill pain, this is its structure. That's its chemical structure, right? It has an oxygen atom, a carbon atom, oxygen atom, and so on. And these are the bonds. This is double bonding, this is single bonding, and so on. Arthur Kelly said, for this, you represent it as a graph in this manner, where you have your atom there, you represent by a vertex. Where you have another atom, you represent by a vertex. Because they are bonded, you join using a line. And this model of a compound then became widely known as the molecular graph. The molecular graph of a chemical compound. The beauty about it is now that we can think of chemical compounds as graph structures. We can think of chemical compounds as mathematical objects. Now, the compounds have been transferred into being a mathematical object. We are now mathematicians, and we can deal with them in any manner that we so wish, in any manner that we so desire. So he actually gave us a very good starting point. So when the chemical compounds are now in our world of mathematics, we can do whatever we want with them. But in mathematics, you know that we start their property. So we can then study the properties of these graphs, these chemical compounds, thinking of them as graphs. We study the parameters of the graphs and we link it back to what we want to find in terms of the chemical compounds. For instance, we then link it back to say, well, if we want potency, if we want toxicity, what are the properties that we desire on this side so that we get this on the other side? One of the indices that has become very instrumental is actually the irregularity index that we spoke about. That is actually quite useful. So remember, our worry has been pain. Some of you may not know this, but in our bodies, we have something that is called enzymes. They have certain specific functions. And one of the enzymes that's important for pain is called the COX-2 enzyme. And this is its cartoon structure. So these things are actually there in our bodies. So the COX-2 enzyme is responsible for inflammation in disease or when we are injured. The COX-2 enzyme is responsible for inflammation, for pain, and for fever. So because it's responsible for that, when we want to worry about killing pain, inflammation, and fever, we should then target COX-2 among others. The best would be to target COX-2 enzyme. So what exactly do? We simply want to find a drug. Remember, these drugs are graphs. We simply want to find a graph that can actually inhibit the work of COX-2. So we stop COX-2 from working, we won't feel pain because COX-2 is responsible for sending messages throughout the body, through to the brain, to tell the brain that, look, we are in pain here. So if we inhibit the work of COX-2, then we've got it. If we inhibit the work of COX-2, then we won't have inflammation because it's responsible for that. So that is where we need to concentrate our efforts on. We need to find, those compounds that inhibit the work of COX-2. So that's our goal. But the question is, how do we measure the extent of COX-2 inhibition by a compound? Remember, when you have a compound, you obviously can't, you cannot avoid talking about quantity, right? So generally the quantity of a compound that we need to stop a biological process by 50% is called IC50 value. Sometimes we use EC50 value, but it's almost the same thing from the mathematical point of view. So the smaller the IC50 value, the better if there are no other side effects. So for instance, Rofucoxb has an IC50 value of 0.53 micromolar, and ibuprofen, I think this is really used a lot, and Panadol used a lot. Panadol has an IC50 value of 25.58 micromolar. Now, our question is, can we design beta COX-2 inhibitors? We spoke about side effects. Can we design beta COX-2 inhibitors? Well, we say, let us think of our compounds as graphs. So we are really asking the question, can we find graphs with beta, for instance, beta IC50 values? But the starting point is, can we get a model, a mathematical model that can predict the what? The IC50 values. And we actually did that, and we managed to produce a model. There you can see our model 2, where all these are graph parameters, and I can tell you that one of it is irregularity index, and that actually managed to predict our IC50 values. So we then took it for a test, and we actually managed to discover 5 compounds using the model. So the model discovered 5 graphs and said, you know what? These graphs here represent compounds which have got better IC50 values, which have got the potence that we want. So the first compound that was discovered is actually an IC50 value of 0.58. The lecoxib is around 0.8. The second one actually has a smaller one, 0.01. The third one, 0.07. The fourth one, 0.0. The fifth one, 0.77. This is the mathematics world. This is so theoretical. We are just talking. So, very nice results. And so what do we do with that? All theoretical. I've got a team of researchers that I'm working with. And this is Dr. Ferreira from MCB here at BETS. And our team of students, we've got postdocs. We also have PhD students, and we also have honors and master's students. They said, look, we are going to take these compounds into the laboratory. We are going to take your graphs into the laboratory to check how they perform. And you can see the results. All the compounds showed approximately 50% COX-2 inhibition, according to what the model had said. Remember, there are millions and millions of compounds. The model selected those, and these are the results. So for compound 1, they actually took 5 concentrations, and the middle one is the one that was suggested by the model. They took these ones and those ones, and these are the results. This is how much they inhibit COX-2. And you can see that it's slightly above what? 50%. So in other words, we are saying, if you take that compound as a drug, assuming that all processes are completed, then you expect that with that quantity, the one that was given in the table, then you expect that you will inhibit the wave of COX-2 by roughly 50%. And so it means your pain will go down by what? By a respective amount. But that is if all processes are completed. So they also checked whether the compounds were toxic or not toxic. And the results are also very good. The compounds are actually non-toxic, which is a very good thing. And as I said, some of the compounds in pain we are taking for animal studies, and the center is actually was so generous to fund that. I also promised that we are going to talk about Alzheimer's disease. This is how our brains look like. I'm not a biologist. I'm a mathematician. So for me, the most important thing is, well, this is the bad side of things, and this is the good side of things. The bad side is your cells are what? Are dying. And you have what we then call Alzheimer's disease. Unfortunately, with Alzheimer's disease, it was noticed that it actually affects over 55 million people globally, which is a very sad thing. Currently, the therapies that are there are not effective, and they've got severe side effects. For instance, you have brain swelling, you may have bleeding, and of course, that will lead to death. And it's very, very expensive, about 25,000 US per year per patient to look after someone with Alzheimer's disease. Now, since 2003, some reported that there have been over 200 investigational programs that are very expensive, that are protracted, and unfortunately, there's no new drug approved so far for Alzheimer's disease. So some people even made huge efforts. They took a number of chemical compounds into the laboratory and they did tests to determine their potence and all those other involved things. And unfortunately, until today, still no best drug as yet, which is a concern. So myself and colleagues, we actually also said, let us attempt to see what's happening. Let us attempt to see if we can make a contribution from the METS models. You can see the names of the colleagues. Prof. Nyabaza is from UJ, our former postdoc. Then we have Prof. Young, we have Dr. Martin, we have Prof. Mpakobanga, and we've got Dr. Ferreira from MCB and so on. So we tried it out and we actually managed to develop something, a model, a mathematical model that for every compound under the sun can actually associate the compound with what we call here a score. And so surprisingly, the score associated to each compound can actually be used by a function, an exponential function, to predict the IC50 value of that particular compound. That is automated. So you can run the mathematical model, screening thousands and thousands of compounds in a very short space of time. 15 minutes, you have done 20,000 compounds. And what is the output? You get the list of the potence of each of the compounds. And you can even ask it to sort that in increasing order. So your best ones will appear first. So this is what we did. And actually, the model identified two compounds, which we called W1 and W4. And with those IC50 values. Remember I said IC50 values and EC50 values almost the same. So we found two graphs that have got really good properties. So for this one, that's 40 nanomolar, that's very small. Even 0.05 nanomolar, that's really very small. So we are saying for practical purposes, once all processes are done, you just need this little bit in order to cure whatever by 50%. That's a very nice measure. Now, the two graphs that were found, W1 and W4, actually went for the wet laboratory experiment. And it was realized that, yes, remember we said that in Alzheimer's disease, your brain cells are shrinking, they are dying. So when they took these to the laboratory, they noticed that those two graphs actually rescue brain cells from dying. They rescue brain cells from dying. And these are the results. You can see that this cell here is what they use in the laboratory to model the brain cells. And these are the compounds, W1, W4. And what they did was they took a chemical compound that kills the brain cells, which is hydrogen peroxide. They poured it in and they killed the brain cells. And they took each out of our graphs in there. And then they could see the brain cells coming up back to life. So these were some of the results that came out of the laboratory as confirmation to the work of the model, and that's a mathematical model. Prof, I thank you for this opportunity. This is the end of my presentation.
Thank you. I see the rest is confidential. Are there questions?
Thank you so much, Prof, for the talk. My first question would be, why do we care that the graphs are irregular or regular in designing the tracks?
Why do we care that they are okay? If you look at this structure, I'll go back to that structure. Yeah, this is the cartoon structure of your enzyme, right? It has sites where your drug goes. These sites have got different shapes, right? Think of a key system. One key may be able to open, but others may not be able to what? To open. A regular key might fail to work. A non-regular key might what? According to some criteria, might succeed in what? In working. So the shape and form of that structure informs us. So that's nature. That is what it is. You will then find out that when you develop the model, regularity will not, always regularity will fail to unlock.
So in most cases, they are irregular.
In most cases, but now it depends with the disease and it depends with the target. So it's not cast in stone. Yes, it might be possible that for some targets, your regular graph might what? Might work. But obviously for, say, Alzheimer's, as I said, and say for pain, as I said, the irregularity index had a hand in it. It's just as good as when you say your body mass index, your BMI thing. It's a function of 2 or 3 things, right? It's the same thing. The irregularity index was one of the independent variables that got into that function.
Let's try some other questions.
Okay. The last one, what can we say about the structure of the eye graphs that you guys found in terms of them being isomorphic, all of them being non-isomorphic, or safety sequence?
Yeah, what I can say is those 5 structures are, yeah, that I can disclose. As Prof said, some of it is confidential. You know, as a mathematician, I would really like to show you the graph. In fact, I can't wait to show you that graph, to show you those 5 compounds. I can't wait. But then we first of all have to patent. So after patenting, it will be all good. Then I can say, Doc, please come to my office. Here are the structures. Yeah, something like that.
Hey. Thank you, Prof. 2 questions. One, are you able to use the same techniques to model toxicity as you have in potency? And the second question is, have you tried any of these compounds on yourself?
Well, I like the second question more. Let me emphasize that when you design a drug, there are clear procedures to be followed. I'm not allowed to take any of these compounds. I'm only allowed to do so when they are registered. But for you to be able to register, you need first of all to do your clinical trial, preclinical trials, of course, after the discovery, meaning to say do the lab work, generate the data, do the animal studies, then you do the clinical trials. Then once approved, then you are allowed to take it. But I have never done that. I've never done that. But there is also a line of inquiry where we say we are repurposing drugs, right? What exactly do I mean? I may actually bump into using these models, into realizing that, oh, okay, you know, this high blood pressure tablet, it works for killing pain as well. You know, things like that do happen. So in that case, it's conceivable that if I identify a high blood pressure tablet as one which also kills pain, right? I can talk to people who have been taking it for high blood pressure and say, do you have pain, right? And they can tell me. But that's when you do what? Repurpose. But I would say of those 5, some are actually for repurpose and some are actually no fail. Oh, sorry, he had another question. Yeah, he had another question. No, I had not answered that. So what our models do is they predict various properties of the chemical compounds. One of the properties is potency. Another property is toxicity.
You do model toxicity.
Yes, 100%. So it depends on what you are using it for, and you then recalibrate it to be alive to what your request is. Remember, when you design a drug, it's not only, say for instance, you are designing a drug, a sex pill, right? You also want to worry about how long will it stay in the system, right? You wouldn't want a 5-minute pill, right? It's something that will, the half-life, you know, something that will be there for only 5 minutes. You wouldn't want that. So there are quite a number of, and you wouldn't want something that's toxic. You wouldn't want to have a heart attack, right? You know, all those things. So our models actually model various properties of chemical compounds.
Measure of the regularity of just constant values and constant thinking for the various things. So that multi-set of values. See what happens. And then the second was much more how do you decide, you know, which
So which one is?
So once you know the structure,
How do we come from the?
How do you know what to put in each of these places?
Yeah.
Oh, okay. Like I said, this is a what?
Toy model.
A toy model. So this is oversimplification of the.
And as I think the regularity is oversimplification, the only parameter.
Yes.
Right.
So we totally agree that these assumptions, some of them are not realistic. But this is work that came through in what, about 1870.
Right.
Yeah. So one thing that I can assure you is the models that we developed are so much different from. This only gave a starting what? I would love, I mean, after patenting, to discuss the details, to say, hey, this is, hey, this is, hey, this is. But now at this stage, before protecting, I can't say more. But yes, I do acknowledge that you can't move from there to there. Yeah. You can also even see that this is a double bond, right? And the edge that is representing that is this one. It's a single what? Edge. And this model is actually looking at this edge as the same as what? That edge. It's the same as this edge as well. So it's an oversimplification of the what? Of the truth. But it only gave us a starting point.
There was an online question. Yes.
So I'll just read it out. So this one is from Mohammed. He says, "Thank you very much, Prof, for the nice presentation. I want to ask what type of predictive models you use for predictions. Are they regression models?"
They are math models. I think that's where I can just end. They are math models. We are still talking about it with our expertise here to see how we can protect that. Yeah. First, after protection, then we can disclose everything.
All right.
Yeah. I'm sorry about that.
There's a whole lot more. So Joel raised a hand. I'll just unmute Joel, then they can ask.
Good morning, Prof. Good morning, everyone. Thank you for the wonderful presentation. I would like to ask, you mentioned CoE-2 enzyme, and you use IC50 to measure maybe it is above 50 or not. Since that is the natural thing in our body, as you mentioned, what percentage is permitted? You say you mentioned 50, a little above 50. So at what range of percentage is permitted that will not hurt or cause harm to the body cells and other things in the body? That is the first question. The second one, I will assume that below 50, no effect will come on the body. I'm not sure if that is right. Is that right or not? Thank you, Prof.
All right, thank you very much for that. The way the IC50s work, it's just a measure, right? It is just a measure. So when we say IC50, what we are referring to is what is the quantity of the compound that you need to get into the body so that you have your 50% inhibition of the biological property or biological process. But it does not mean to say for us to cure someone, we need that quantity. This is only for our information to say, if we put in this, then we can lower the burden by 50%. If in order to be successfully healed, you need the quantity for 100%, then it's simple mathematics to get the right dosage. So that then takes us to calculation of doses of how much exactly we need. But of course, that will be based on the IC50 value. So if your IC50 value is, let's talk about liters that we are all happy with. Liters. If you need 50 liters to be cured, right, there is a problem. It's just too much, isn't it? Why? You might even have side effects because it's already what? Too much. It's not permissible. So what we desire to have is a situation whereby you have as little dose as possible, giving you 100% clearance of the burden that you have in your body. So it's not a function of one thing. It's a function of several things. Yes, you have taken this quantity and we know it clears you maybe by 50% or by 100%, but is that quantity then not going to become harmful to your body. Because a little bit of something may even be toxic to your what? To your body. But you can put more for some other different compounds and you won't even harm your body. But you may probably not then be able to clear the what? The burden. So it's quite a number of aspects that are considered outside even of the potents. But of course, potents is the main issue.
We will now take one from Augustine Nangi that raised their hand.
Hi, Prof.
Prof, good morning.
Hi, good morning. Great talk. Thank you very much. This is a wonderful team and a wonderful ambition, if I may use that word. Obviously, I presume that the dream of this whole project is to one day be able to play a major role in manufacturing drugs that will actually help people in the street. It's a big dream, even though you've mentioned answered a bit of what I wanted to ask. But what about in the name of quality control? The entire research, one would presume that it is done, with kind of collaboration or with pathologists, chemists, just for quality control purposes. And also, what about how far has this been, kind of published towards achieving this kind of goal? Because ultimately, for that ultimate dream to be achieved, quality control is essential. What do you think?
Yes, Prof. What you are saying is very, very important, and thank you so much for the kind words. What you are saying is very, very important. As mathematicians, we play our part in the collaboration. I must say the collaboration has a lot of expertise. When I talk about getting into the laboratory, I will go there to observe and see, but I know nothing. So my colleagues in biology, my colleagues in biochemistry will then do their part. As you saw some of the graphs, they came from the biologists themselves, the biochemists, saying these are the results that we got from their field. They are experts in that field. When it comes to these compounds, there is also talk with the chemists, not the biochemists and the biologists, with chemists. They are the ones who know how to synthesize the compounds. They produce the compounds, but basically we go there and we tell them, we want a compound that has this structure. We want a compound that is this particular graph, and then they create it. After creating it, then we take it to the biologist and we say, please do the test, and then they will do the test. But in this research, we actually have physiologists as well. When you take your compounds for animal studies, you no longer really require a biologist or a chemist or a mathematician. You now need people who are knowledgeable in that area. So they take the research further, and they are the ones who will do the animal test and not us. So that is cooperation, that is joint work, that is joint research. And if you look at all this that I'm mentioning, there is high appetite then for getting postgrads to be involved in that. I clearly know, maybe I've lost count, but we have a number of postgrads who are working on it. And these are research problems, practical research problems that are now getting into their dissertations, and those are the research problems that they work in. Then we also have pharmacists, and these pharmacists, some are national, some are local, some are international. they are also working together with us because they've got knowledge about these drugs. And so we don't work in a vacuum. We have collaborators. But in terms of the quality, remember the patenting process, it's just as good as you are publishing something. You don't just get a patent, and just like in publications, you send your work to the journal will send to reviewers will do their quality control measures. The same applies with patents. It's not like you wake up and say, oh, I now need a patent, and they, oh, please patent. No. It's reviewed, and the quality control is also done. And then, oops, you now have your what? Your patent. So the quality control is definitely going, but unfortunately, before we could publish it, first of all, we need to what? To patent. So that's why we then don't disclose a lot, so as not to jeopardize our chances of what? Of patenting. You can't patent something that is common knowledge. So if I publish now, the patent people will say, ah, this is now public knowledge. Why would you want to claim that it is yours? When it's what? It's already, in the public domain. So then we don't want to jeopardize that.
Last one is from Mabab Boniface Matad. I hope I'm pronouncing that well. So wonderful presentation, Prof. I would like to find out whether there is any reason of having 8 variables while you model IC50.
All right. Thank you so much, Prof. Baba. Well, what I can say, this is nature, right? Sometimes it's possible to model using one single variable. Sometimes it's possible. We have had a lot of cases where we could just use what? One single variable. But it depends on the context where you are. Sometimes even the 8 will be a small number because there will be various aspects to be looked at. But for these particular models, because we have not patented, it just remains what? Know-how. I can't disclose a lot outside of what? Outside of that. I hope that clears the air.
Yeah, it's clear, Prof. It's clear.
Thank you. Thank you, Simon, and thank you to the audience for a very interesting presentation, and we wish you great prosperity in it. We hope that one day some pharmaceutical companies will buy your patents and produce these new drugs in large quantities.